All right, good night, everyone, and welcome to our last webinar of the Beef Reproduction Task Force this spring. We are very pleased with the results. It's been a lot of, a lot of good talks and a lot of good attendance. So we've been very pleased with the results of this webinar series, and we're planning to continue the series next spring. So I want to thank everyone that has spent the time and join us every third Wednesday of the month, and we plan to keep that going for next year. Our last speaker of the series, last but not least, Dr. Pedro Fontes. He's a good friend, and we've known each other from way back in Brazil and then from grad school. But Dr. Pedro Fontes, he's got his DVM from São Paulo State University in Brazil, then got a master's from the University of Florida and a PhD in beef cattle reproduction, reproductive physiology from the Texas A&M University, and is currently an assistant professor at University of Georgia in the Department of Animal Science. So Dr. Fontes is going to speak to us today about pregnancy diagnosis and new technologies on pregnancy diagnosis. So we're really looking forward to your talk. And Pedro, the floor is yours. I forgot, before I turn to you, we are live on Facebook, and also if you have ever missed one of our webinars, they're all recorded and they're available on our YouTube channel, the Beef Reproduction Task Force YouTube channel. And this webinar will also be available tomorrow morning on YouTube. And at the end of the webinar, we're going to have some questions and question and answer time. So please use the Q&A tab of Zoom to send your questions. Okay, Pedro, now the floor is yours.
All right, Vitor. Thank you for the introduction. As we go through here, let me know if you have any issues listening to me or anything like that. But let's go ahead and get started. First of all, good evening, everyone. Thanks for joining us today. I think that's an interesting topic that I really enjoy talking to producers about. Different pregnancy diagnosis technologies that are available. And And just as sort of a brief introduction, as Vitor mentioned, I went to vet school in Brazil, so I graduated from São Paulo State University. Then I went to grad school at the University of Florida, as well as Texas A&M University. So I got my PhD from Texas A&M University. And I have recently started here at UGA, at the University of Georgia in 2020. And I'm really happy with the invite from Vitor, because we're going to be able to share some of the work that we started doing here, looking at some new technologies to diagnose pregnancy. More specifically, we've been working with color doppler ultrasonography. So trying to diagnose pregnancy a little bit earlier than we currently do with our conventional methods. But for our conversation today, I want to go over not only the new technologies that are rising, but also, and some of the work we've been doing, but also the more traditional methods of pregnancy diagnosis, right? So before we go ahead and get started, I want to have a brief introduction just to kind of get our conversation started with regards to beef reproductive efficiency. So here I put together some data that I got from FAO, and I organized that data based on different decades here that you can see here in the Y and the x-axis of this figure. So here on the left, we have the number of cattle slaughters here in the US, and here on the right, we have the amount of beef that we produce. And then you can see that as we move from the 60s all the way into the last decade, so 2010 here represents all the data collected from 2010 all the way to 2020. And what we see when we look at those overall industry trends is that we have experienced a decrease in the number of animals that we've been slaughtering over the years, and that decrease is about 16%. And what's interesting is that in the same interval of time, we're able to increase beef production by about 30%, meaning that we're basically producing more beef with fewer animals, and really highlighting that we're getting more efficient with our process of producing beef, right? There's a lot of factors that have been driving those differences in efficiency, but definitely one of them has been the changes in carcass weight, right? So when we evaluate the changes in carcass weight over the last 50 years or so, we can see that we increased carcass weight by an average of about 50%. Basically meaning that to produce the same amount of beef that we get from a carcass between the years of 2010 and 2020, in the 1960s, we had to slaughter 1.5 animal, right? So meaning that we really changed the characteristics of our animals. And so when we consider all that and we think about some of the future directions of the beef industry and some of the things that I think are relevant for this group here, we know that it's very unlikely that our herd numbers are going to increase. So it's likely that we're going to have our herd numbers stabilize or potentially decrease our animal numbers. And as we look at the carcass data that I showed you in the previous slide, it's very likely that really rapid increase is going to probably plateau, right? We know that increase, it's not sustainable on the long haul looking forward for several reasons, right? We know that we will have to have some serious adaptations in our packing plants to process carcasses if they start getting too heavy. And we also know that there's several implications to the cow herd as we start thinking about cow maintenance and things like that. But besides of that scenario, the specific scenario here, we know that the demand for beef, and not only for quantity, but also for quality of beef, is likely going to continue to grow as we move forward, right? And, you know, as a reproductive physiologist, the way that I like to approach this is to think that we're going to, as cow-calf producers, we're going to be playing an important role on that overall increase in efficiency that we're going to be observing in the future, right? So if we want to be able to meet that demand for quantity and quality of beef, we're going to have to improve our efficiency in the cow-calf segment of the industry, right? And by improving efficiency in the cow-calf segment of the industry, I'm talking about producing more pounds of calf per cow exposed in the breeding season. And that's pretty relevant as we start talking about the use of reproductive technologies in the United States. And this is a survey that was put together by the USDA, where we have different herd size here on top. So we have small, medium, and large operations. And then we have different reproductive technologies here on the left, okay? So we have resynchronization, artificial insemination, embryo transfer. But what I want to draw your attention to here is the three reproductive technologies here at the bottom, because they're relevant to our discussion today, right? So when you look at the rate of relatively simple reproductive technologies, right? Pregnancy diagnosis through rectal palpation or ultrasound, and more recently blood tests. The rate of the percentage of our operations that are adopting these technologies are considerably low, right? And what we see as well that it's interesting about this table is that as we increase the size of the herd, we also see an increase in the rate of adoption of these technologies as the herd increases, right? Victor, just want to double check if everybody's seeing my slides. I'm getting some messages here in the chat. Can you see my slides okay, Victor?
Yeah, Pedro, it seems that some people are getting your video instead of the slides. I think if your video is spotlighted, so I'll try to fix that, but please keep going. We'll fix it.
All right, sounds good. So yeah, so we know that and when we look at this data, we see that as the size of the operations increase, the proportion of producers that are utilizing reproductive technologies also increase, right? And as we start thinking about the reasons of that, there's likely several reasons for that. But one of that is likely that the percentage of the annual income of those families that run those larger operations, likely a greater percentage of their income is coming from their cattle operation, right? And those folks are usually aware of the benefits of the utilization of these technologies. So they're taking advantage of these technologies. So as you think about improving efficiency moving forward, hopefully in a few years from now, if we conduct a survey again, hopefully we'll be able to see an increase in the incorporation of these technologies. So why should we perform pregnancy diagnosis? Why should we worry about running cows through and collecting that information? So there's several reasons, right, why we want to collect pregnancy diagnosis information, right? And we don't only want to collect that information, but we also want to use that information to actively make management decisions, right? So as we know the pregnancy status of our cow herd, we can make changes to our nutritional plan, but not only that, right? We use that pregnancy status information to make marketing decisions, right? So if you think about traditional cow-calf operation, usually about 15 to 30% of their income are going to be coming from cow-cows. So having the pregnancy diagnosis information and establishing our culling criteria based on lack of reproductive efficiency, it's substantially important, right? Not only that, we know that having that information together with knowing, that there are seasonal variations in cow prices and combining that with different prices in commodity can really help producers develop strategies with regards to their with regards to the cows that they're culling, right? There's several options and several things we can do, ranging from adding weight to our cow herd or immediately marketing them, depending on the market situation. So having that pregnancy diagnosis information is really important. And as we start talking about, evaluating reproductive efficiency, so a couple of days ago, we were preg checking some of our cows that we transferred some embryos. So I really want to evaluate how we're doing with regards to the use of these reproductive technologies, either if we're talking about embryo transfer or artificial insemination, or whether we're trying to evaluate the performance of a specific sire from a fertility standpoint. So there's a lot of value to have that pregnancy diagnosis information. And usually when we think about the consequences of neglecting the importance of pregnancy diagnosis, if we fail to detect those open cows, we're going to be holding cows that theoretically shouldn't be kept in our herds, right? So we will ultimately increase the number of animals in our operation. And as we increase the number of animals in our operation, we also increase the expenses associated with cow maintenance, right? Which ultimately increases our production costs. Not only that, as I mentioned earlier, those cows usually represent about 15 to 30% of our income, right? So if we fail to market those cows, to properly recognize those less fertile cows and cull them and market them, we're going to ultimately decrease the income that is coming from those cows if we're not utilizing pregnancy diagnosis. Not only that, by doing so, we're going to fail to cull some of our less fertile animals. And we know that as we keep those less fertile animals on the long haul, we're going to decrease reproductive efficiency and reproductive performance over time. So before we go ahead and start talking about specific methods for early pregnancy diagnosis, I just want to talk a little bit about what's going on, right, during early pregnancy. So here I have a figure of a representation of some of the physiological events that are happening during early pregnancy. So here we have the uterus, actually the uterine horn of a cow, okay? And then we have the oviduct here, and then these yellow structures are the embryo here, and here, more towards the left side of the diagram, we have the representation of organoocyte, right? So we know that when a cow ovulates, that oocyte is going to arrive here in the oviduct, and hopefully when it's kind of halfway through that oviduct, that oocyte is going to get fertilized, right? And after fertilization, we have the formation of an embryo that is going to arrive in the uterus, usually around 5 to 6 days after ovulation, right? And for those of you that have done some embryo transfer work and have seen folks flushing cows, and you get a chance to look at the scope while the embryologist was sorting those embryos, you've probably seen a figure similar to this one. So that embryo arrives in the uterus in a spherical shape. So this is day 7 embryo. And then as you can see, around that embryo, there's a protective layer, which is what we call a zona pellucida, okay? And as we progress in gestation, that embryo is going to hatch from that zona pellucida. So this is a electron microscopy figure, which is basically a fancy way to visualize those embryos in a research setting, where you have that spherical embryo hatching from that protein protective layer from that zona pellucida at day 8 of development. Okay, so as we progress in pregnancy, that embryo that was once spherical starts elongating, gradually elongating, and it acquires this tubular form. So this is a electron microscopy as well of an embryo on day 13 in a tubular shape. And then as we progress in pregnancy through day 14, that embryo keeps elongating all the way until day 17, here where we have a flush conceptus in what we call a filamentous form, okay? So you can see that this conceptus and that embryo undergo some dramatic morphological changes, and not only morphological changes, we know that they undergo a series of functional changes. And every time we're talking about some pretty substantial changes in biology in early pregnancy development, we're talking about the very high probability of things going wrong, right? We know that there's a lot of pregnancy loss that occur not only from day 7 to day 17, but even before that now. And also after day 17, as that embryo starts attaching and forming the placenta within against that uterine lumen here that we have represented here on top. So there's a lot of things that again go wrong. There's a lot of research efforts that folks have been putting on trying to understand what are the factors that regulate the developing of those embryos, and what can we do from a management standpoint to optimize all these events to maximize fertility. But what I want to share with you now, it's a meta-analysis that was done by Dr. Polar's group, where he basically characterized what's going on in the beef, in our beef cattle population with regard to those pregnancy losses that occurred not only during early gestation, but also towards more advanced stages of gestation as well. The take-home message that I want you to have at this slide is that as gestation progresses, a lot of pregnancy loss occurs, right? So if you look during early gestation here, and this is a series of studies that they compile in this meta-analysis where folks were going there and actually collecting the reproductive tract of a cow, you know, bringing to a laboratory and flushing those tracts and visualizing the percentage of cows that actually had an embryo-like structure during different stages of gestation, as well as data from the field where they go there and perform pregnancy diagnosis with ultrasound and later stages of pregnancy to really characterize when those pregnancy losses are occurring. So basically, if we go here and you synchronize a group of cows in a fixed time AI settings and artificially inseminate them, we are here at 100% of the cows being serviced, right? And as we move forward in gestation, pregnancy loss start gradually occurring, right? So for example, based on that meta-analysis, if you go here day 10, let's say we develop the best pregnancy diagnosis method possible that can detect 100% of the cows pregnant here at day 10 of gestation. If you think about the real value of that pregnancy diagnosis for a cattle producer, it's not really valuable, right? Because naturally, some of these cows are going to experience embryonic mortality here as gestation progresses. So as we talk about trying to diagnose pregnancy early, this is something to keep in mind, okay? And you know, for better interpretation of this idea of pregnancy loss, what folks have been doing in the literature is dividing the pregnancy loss in different periods. And usually when I look at most of the beef research looking at pregnancy loss, usually they classified the pregnancy loss that is occurring from day 0 all the way to day 30. So we're doing our first pregnancy diagnosis as early embryonic loss. And then when we're talking about the pregnancy loss that are occurring between day 30 and day 60, or day 30 and day 100, depending on the experiment. Usually we're talking about what we call late embryonic or early fetal loss, okay? And basically, you can see that the rates of early embryonic mortality are substantially high compared to the rates of late embryonic mortality. And this is going to change a little bit here on the late embryonic mortality side of things. If we're talking about dairy herds, that's a little bit different. Those rates tend to increase. Not only that, if we're talking about folks that are utilizing in vitro embryo production, we know that the rates of embryonic mortality are a little bit higher here as well. But something to keep in mind is that, you know, those cows that experience early embryonic mortality, they're very likely going to rebreed within the same breeding season, right? However, those cows that experience late embryonic mortality are those cows that, you know, kind of break our hearts. You know, you go there and you synchronize your cows and you use a really high quality sire, produce a really high quality, you think you're producing a really high quality offspring. You go there at day 30, you back check that cow is pregnant. But then you run that cow again 30 days later or at the end of the breeding season and she lost that pregnancy, right? So that's really frustrating, you know, from a producer's standpoint. But not only that, the economic consequences of those pregnancy losses are pretty substantial as well. So when you start trying to, when you start looking into some of the research that is doing comparing different methods of pregnancy diagnosis, you see a lot of different terms that are quite complicated. So for our conversation today, I'd like to use two main terms as we start describing the accuracy of different pregnancy diagnosis methods, right? And these terms are false positive and false negative, okay? I'm not going to talk about sensitivity, specificity, and positive predictive values. I'm going to be focusing mostly on false positive and false negative terms. And basically what I mean by a false positive is when we go there and we have a cow that is open and we use a specific pregnancy diagnosis technique, and we go there and you call that cow pregnant, but she's actually not pregnant, right? So that would be what we call a false positive. Whereas a false negative would be a cow that we know that she's pregnant, right? But we go there and we use a specific method to diagnose pregnancy, and that method tells us that that cow is not pregnant. So that will be what we call a false negative, right? So as we think about an optimal pregnancy diagnosis scenario, we want to avoid both false positives as well as false negative results, okay? With regards to false negative results, these are likely more important as we think about the management practices of a beef operation, right? So if you're resynchronizing cows, we don't want to have false positive results, false negative results. And also, if you're thinking about marketing cows, we also cannot afford having false negative results. So these are things to keep in mind as we move forward. So let's jump in and start talking about some of the current methods that are available. So likely the most commonly utilized method for pregnancy diagnosis is rectal palpation, right? So when we're performing pregnancy diagnosis with rectal palpation, we're going to bring the veterinarian in. And one of the things that I want to bring up before we start diving into these different methods is that regardless of the method that we're utilizing, you know, we really, it's really important that producers work with their veterinarians, right? To come in and provide that accurate result that you can really trust and you know, develop that relationship with their veterinarians, which is really important, right? As we talk about these different methods, make sure you're working with their local veterinarian to bring them in and perform those pregnancy diagnosis results. So as we think about rectal palpation and compared to some other methods we're going to talk about, rectal palpation usually is performed a little bit later, right? We've got to wait until around day 40 of gestation to start being substantially accurate with the rectal palpation. Again, it requires a trained personnel to perform the test, so we usually recommend that you work with your veterinarian. That's really important. And one of the good things about performing rectal palpation compared to other tests, so for example, the blood-based pregnancy diagnosis, is the fact that we get chute-side results, right? We go there and we palpate our cows and we can make a decision right there whether we're going to sort that cow, we're going to send her to market, or whatever we want to do. Another positive thing about the rectal palpation method is the fact that, you know, depending on how trained the veterinarian that's performing those diagnosis is, they can detect quite a few pathologies such as, you know, mummified fetus and things like that. And that's definitely a positive of rectal palpation. And so basically when the veterinarian comes in and he's rectally palpating our cows, he's looking for some specific landmarks. So here, these are some figures from the DRAWS project. So here I have a pregnant tract, reproductive tract on the left and an open reproductive tract on the right. So here you have the different uterine horns, and I want to draw your attention to the fact that these uterine horns are pretty symmetrical, right? And here, when you go to this pregnancy here and this pregnant tract, this is probably around day 50, day 60 of gestation. And you can see that there's a clear asymmetry between the horns. So whenever your veterinarian is in and he's performing that rectal palpation, he's trying to palpate those both horns and trying to feel for fluid inside the uterine horn, but also that asymmetry between the two different uterine horns. Some other things that we're going to be looking for as well is, of course, the fetus itself. But not only the fetus, actually the placenta, right? So this is a pregnancy that was dissected out of the uterus, and you can see that the fetus is free floating here inside, it's floating here inside that placental structure here. And you can see this button-like structure. This is what we call the cotyledons, because here we have only the fetal component present here. And the cotyledon together with the caruncles form what we call the placentome. So whenever your vet goes in, he can feel some of those placentomes depending on the stage of pregnancy and use that to diagnose pregnancy. Another method that it's commonly utilized in beef operation is ultrasonography, and I'm calling conventional ultrasonography here because we're going to talk a little bit about color Doppler ultrasonography as we move forward in the presentation. And you know, as we think about the advantages of utilizing conventional ultrasound, we can detect pregnancy a little bit earlier. In heifers, we can go as early as day 26. In cows, as early as day 28, depending on how trained the person is, potentially even earlier than that. Not only that, but with the ultrasound, we can have a pretty good idea on the viability of the pregnancy, depending on the stage of pregnancy as well. But not only that, we can pretty accurately age that pregnancy, a little bit more accurately with ultrasound than with the rectal palpation itself, mainly when we're talking about early pregnancy diagnosis. Not only that, we can also determine the sex of the fetus depending on the stage of the pregnancy that we're evaluating those cows and those fetuses. Cost will vary similar to what happens in the rectal palpation. And from how those, when you think about how those ultrasound examinations work, so basically we have an ultrasound machine here on the right, and then you have a transrectal probe. So we're going to introduce that probe in the rectum, and the probe is going to send ultrasound waves. Those waves are going to hit an object. In our case here, we're talking about the reproductive tract. That object is going to bounce waves back. They're going to be received by that same probe, right? So depending on the length and the characteristics of that wave that bounces back, that receiver will interpret those waves and translate that into an image that has different shades of gray. And basically those waves are going to be changed based on the density of our object here. So if you're talking about a fetus that has bones and it's pretty dense, let's call it like that, the image that we're going to be visualizing in the ultrasound is going to be bright, right? Whereas if we're talking about a tissue that is less dense or something that is not dense at all, like water, the type of image that ultrasound is going to generate is going to be a little bit darker, right? So this is an example of some of our of a video that we recorded recently here in one of our preg checks. This is a day 85 or so pregnancy, and here you can see the skull of the fetus, the head of that fetus, surrounded by this black image here, which is the fluid contained within the uterus, right? So that the fetus is floating within the uterus, and then we're visualizing fluid as well as the fetus itself. So that was a day 85-ish pregnancy. And now I'm going to show you an early pregnancy. So if we're going around day 28, day 30, this is something, this is what you're going to be observing. So this is a cross section of the uterine horn. We have a pocket of fluid right here. And then you can see a button-like structure popping up, which is that developing embryo here with about 29 days. That's when we perform the specific pregnancy diagnosis here, okay? So once you know the person gets trained on how to visualize in these images, it gets quite easy to interpret what's going on and recognize those pregnancies, as long as you have substantial experience palpating cows, right? So that's why it's important to bring someone in that has those skills, and that's why we encourage everybody to be working with their veterinarians. One of the things that I want to show you that I thought it was cool, we're recently doing some final preg checks from one of our fall breeding herds, and this is a pregnancy with about 110 days, and this is a cross section of the thorax of a calf. And just to highlight the quality of the ultrasound machines that are available now for us to utilize and how detailed the images are getting. So here I'm going to play that video, and I want you to draw your attention right in the middle of the screen. You're going to be able to see the heart of the fetus beating. But not only that, you're going to be able to also see the different chambers of the heart. So if we pause our image here, you can see the different ventricles, and you can not only see the ventricles of the heart, but you're also going to be able to see the valves of the heart as the heart of the fetus beats. So pretty interesting to see how accurate our images are getting, and the kind of quality of ultrasound equipment that we can utilize to generate some pretty trustworthy results with regards to pregnancy diagnosis methods. So not only that, through transrectal ultrasonography, we can pretty accurately age pregnancy. So this is a couple of images from fetuses at different stages of gestation. So here we have a day 53 pregnancy. Here we have a day 84 pregnancy. And as you can see, they're pretty substantial differences. Right here is just the head of the calf, and here you can see the whole fetus fitting within the screen. So whenever you bring your veterinarian in, they can go in and pretty easily distinguish those pregnancies and be able to age those pregnancies pretty accurately. Not only that, if they come, depending on on the timing in which those pregnancy diagnoses are being performed, you're also going to see, we're also going to be able to distinguish male and female fetuses. Okay, so now let's switch gears and talk a little bit about the blood-based pregnancy diagnosis. And usually when you talk about the blood-based pregnancy diagnosis, folks will come and ask, what are we detecting, right, in those blood-based pregnancy tests? And usually people wonder if it's the same thing as we detect in humans tests that you can buy at CVS or at Walgreens, and the reality is not. So we're detecting something different here. So when we're talking about pregnancy detection in cows with blood, through the blood test, we're talking about the quantification of what we call pregnancy-associated glycoproteins. So this is an image of a pregnancy here. Okay, so you have the fetus here with the different layers of the placenta. Here you have the amnion, the chorioallantois here. But what I want to draw your attention here is to these button-like structures. This is what we call the placentomes. So if you were to grab a placentome from a pregnancy like this and, you know, cut it in half and bring it into the lab and look at it into the microscope, you would see something like this diagram here on the right, where you have a layer of fetal cells on top here represented in maroon, and a layer of maternal cells here represented in yellow. And then in specific stages of gestation, we're going to see that some of these fetal cells will start differentiating, okay? And some of these differentiated cells, they will migrate from the fetal side of the placenta into the maternal side of the placenta. And once they get into the maternal side, they'll tap that basal membrane here, and they'll secrete some products into the maternal stroma here in the uterus. And some of these products will make their way into the maternal circulation, and pregnancy-associated glycoproteins is one of them, okay? So we can go there and collect a blood sample from a cow, from the jugular vein or from the coccygeal vein in the tail. And we can take that sample to a laboratory and quantify the circulating concentrations of those pregnancy-associated glycoproteins, which again, it's coming from those cells that are derived from the fetal placenta. So serving as a biomarker for pregnancy. So here's some of the research from Kai Poller's master's work when he was still in Missouri with Dr. Smith, and here we have the concentrations of PEGs at different months of gestation, okay? And you can see that as gestation progresses, the concentrations of PEGs will gradually increase, right? Not only that, if we were to zoom in during early gestation here, this is what I got here in this figure at the bottom. So we have the circulating concentrations of PEGs at different days of gestation here, starting at day 24, going all the way to day 60 of gestation. And usually at day 24 is when we see the first increase and the first significant difference between pregnant and non-pregnant cows with regards to the circulating concentrations of PEGs, okay? So we can actually go in here at day 30, 28 and day 30 and collect a blood sample and send it to a laboratory and quantify those PEGs and determine which cows are pregnant and which cows are open. So there's three different companies working with blood-based pregnancy diagnosis. So we have the product line from IDEX, Bioprine has their own assay, as well as Genex have their assay. And basically the way that these assays work is these are antibody-based assays. This use technology called ELISA, where basically you utilize antibody to recognize those PEGs in the circulation of the cows. And then based on the recognition of those circulating pegs, we determine whether cows are pregnant or not. And what we get asked a lot is there a better kit, or is there a kit that works better? And the reality is there's a lot of research backing up each one of these technologies. And you know, even though they use different antibodies, and that's pretty much the main difference between them, all these tests can pretty accurately detect pregnancy and yield some pretty reliable results. One of the things that was one of the withdrawals of these blood-based pregnancy diagnosis is the fact that we had to ship those samples to a laboratory. More recently, IDEX, probably a couple of years ago, made it available in the market, a chute side blood test, which is basically a blood test that you can perform chute side. It takes about 30 minutes. Basically, we have a series of steps from dispensing the sample into specific wells to adding specific types of reagents, and those samples will go through a series of washes. And then at the end, you're going to see a changing color in these different wells. So each well represents a cow, and depending on the change of color that you observe in the well, you determine whether cow is pregnant or not. And this is, you know, it's a little bit, it's not super easy, so you need to be really organized to get those going and to get those done. Make sure you're not getting samples mixed here as you start pipetting these into the plates. So these are things to keep in mind as you think about potentially utilizing these technologies. And again, you'll really benefit if you decide to utilize a technology like that, working with your veterinarian, let them guide you through the process of utilizing technology like that. So how accurate are these commercially available blood-based pregnancy diagnoses? And when we look across the literature, the accuracy of those tests are really good, right? So these are two independent peer-reviewed publications with more than 90% accuracy for these blood-based pregnancy diagnoses. And usually, you know, when we talk about 97 or 93% accuracy, folks will go back, and these are preg checks performed, blood samples collected around day 28, day 30 of gestation. Folks will come back and say, well, why it's not 100%, right? And there's a variety of reasons why it's not 100%. And one of them being embryonic mortality. I'm going to show you some data for that. And the other one that it's important, it's cows with early days postpartum. So if we get a group of cows that we breed them when they are early in their postpartum period, what will happen is, as you remember from that figure that I showed you before, the concentration of these pregnancy-associated glycoproteins in the end of gestation is substantially high, right? So once these cows calve, it takes about 60 to 70 days for those cows to clear those pregnancy-associated glycoproteins from their circulation. So if you breed cows really early in the postpartum period, and we collect those blood samples a little bit earlier than we normally do, what might happen is we'll be detecting some of the pregnancy-associated glycoproteins that are coming from the previous pregnancy. So that ends up leading us to have some false positive results, which are not the end of the world, but you know, if we follow the recommendations from those companies with regards to the timing for postpartum period, we can really mitigate those false positive results. So, you know, I mentioned the idea of embryonic mortality and pregnancy-associated glycoproteins. So this is some other research from Dr. Poller's group, where basically we have two groups of cows here, cows that maintain pregnancy and cows that experience what we talked about, what we define as late embryonic mortality. So basically here, we're talking about cows that were diagnosed as pregnant via ultrasonography here at day 30. And then when we ran those cows through again at day 60, day 100 of gestation, those cows lost that pregnancy at day 30, even though they had a healthy fetus embryo here with a heartbeat and fluid, and you know, were considered pregnant through the ultrasonography. So, and what's interesting here is that, you know, when we collect these samples, and we did the same thing here in Georgia, but when you look at Kai's data, here are samples collected at day 30, okay, and those cows are pregnant when we're collecting those samples, but if they're going to experience embryonic mortality, they already have lower circulating concentrations of PEGs. And we observed something really similar here in one of our studies that we're running right now, and these are some preliminary data, but also those cows that experience late embryonic mortality, at day 29, when they're still pregnant, on average, they had lower circulating concentrations of PEGs. And there's some work being done with regards to trying to predict pregnancy loss through the utilization of those pregnancy-associated glycoproteins. But this is, at least at this point, not at a stage where we can utilize that information commercially. What we do know is that those cows are coming in and they have lower circulating concentrations of PAGs. And I mentioned earlier that we can see an increase in circulating concentrations of PAGs at day 24. So can we accurately go there and collect a blood sample at day 24 and evaluate if cows are pregnant and accurately determine pregnancy status? And so this is some research out of Texas A&M University as well. Here we have day 24 circulating concentrations of PAGs for cows and heifers divided in pregnant and non-pregnant animals. And you can see that the pregnant ones do in fact have higher circulating concentrations of PAGs at day 24. However, when you try to utilize that information to actually predict their pregnancy status at day 30, to figure out whether they're going to be pregnant at day 30, comparing to an ultrasound at day 30, which is our gold standard method, right? We have some false positive results, which are likely driven by some embryonic mortality that occurs between day 24 and day 30. But not only that, we have some false negative results. So, the reason why I want to bring that up is that it is really important as we think about utilizing those blood-based pregnancy diagnoses to follow the recommendations, right? If we try to collect those samples a little bit earlier, it's more likely that we might run into issues. So make sure you follow the recommendation of whatever blood-based pregnancy diagnosis you're utilizing, okay? So from a practical standpoint, if we consider all that and we're thinking about scheduling pregnancy diagnosis, if you're thinking about a scenario with 75-day breeding season, we can, after we remove the bulls, 30 days later, run cows through, perform a transrectal ultrasound or collect a blood sample, or potentially wait 40 days and perform rectal palpation. And by doing so, we're going to be able to distinguish pregnant from open cows and figure out potentially calling decisions based on that information. But not only that, if you're thinking about an operation that has a longer breeding season, let's say 75 days or longer, and you want to not only distinguish pregnant and open cows at the end of the breeding season, but also recognize females that are pregnant to AI and differentiate them from the ones that are pregnant by natural service, ideally you want to go in and perform a pregnancy diagnosis earlier during your breeding season, around day 30, day 40 of gestation, and recognize those ones that are pregnant, and then perform a final pregnancy diagnosis to figure out which ones got pregnant by the cleanup bull. One way to do it for the operations that have a short breeding season and want to distinguish pregnant and open cows, but not only that, also distinguish those AI pregnancies. When we're considering operations with a short breeding season, we can come here in and perform a single transrectal ultrasound around 90 days after breeding, in this example of a 60-day breeding season. That will give us 30 days between the removal of the cleanup bulls in our pregnancy diagnosis, which will allow us to differentiate AI and cleanup bulls through the ultrasound here. So that's another option as well as you're thinking about, if that information is important for you. So now let's move forward and talk a little bit about how we can utilize strategically some of these pregnancy diagnosis technologies in the context of resynchronization, right? So for folks that are utilizing astrosynchronization and fixed time AI in traditional, systems, most of the folks that are utilizing AI are exposing cows to one round of fixed time artificial insemination in the beef cattle population, right? Usually those cows are exposed to an astrosynchronization program, and then we go there, artificially inseminate those cows, and about 10 to 14 days later, we're going to turn in a cleanup bull, right? And there are several benefits for utilizing artificial insemination, but one of the main ones is the fact that we can capture some value from those pregnancies that are generated through AI, right? So how can we potentially maximize the number of AI pregnancies in our operation? So there's several different ways that we can do that, and one of the ways that we can do that is by performing heat detection after our first round of fixed time artificial insemination. And this is something that we do in some of our herds here at the University of Georgia, mainly with our replacement heifers, because it's easier to perform that heat detection. We usually perform that heat detection between 15 and 25 days after breeding. And then we will AI those heifers following an AM/PM rule, and we usually get a pretty good pregnancy rates and utilizing this approach, and we really increase the number of pregnancies that we're generating through AI. But not only that, we know that that process of heat detection can be a little bit challenging depending on the size of operation and the logistics of each operation. So we see some operations, and we tend to do that as well in some of our operations, is to utilize resynchronization, right? So we go there, expose cows to one round of estrus synchronization followed by fixed time AI. We go there and perform an ultrasonography examination around day 28, day 30 of gestation. Then we get our pregnant cows, and we don't do anything with the pregnant cows, and our open cows get exposed to a resynchronization program, right? So basically, those cows will get a CEDAR in, a GnRH, and follow a regular synchronization protocol. In this example here, I have the 7-day co-sync plus CEDAR protocol. But something that has been done recently, probably over the last 10 years from a research standpoint, and there's some operations now utilizing that commercially, is starting to resynchronize cows while they still have an unknown pregnancy status, right? So basically, you go there, expose your cows to a regular synchronization program, fixed time artificial insemination. Then at day 21 here, in this example, you go there and start resynchronizing all your cows, give a GnRH injection, put a CEDAR in. And when it comes time to give a prostaglandin injection, which would theoretically abort a pregnancy, we go there and remove that CEDAR, perform a conventional ultrasound, and we detect the pregnant cows. The pregnant cows are let go. The open cows are going to receive a prostaglandin injection and then be fixed time artificial inseminated. So by doing so, we can decrease that interval between our artificial insemination events. So one thing to notice here is that what's driving how early we are rebreeding those animals in a fixed time AI setting, right, in a resynchronization setting, is our ability to detect pregnancy earlier, right? Because we're limited to day 28 of gestation here, we cannot decrease that interval between the two breeding events. And this is something we've been trying to tackle based on some research that it's done in Brazil, and we're trying to apply those results here in the US. So, and that's through the use of color Doppler ultrasonography. So what is the rationale for utilizing color Doppler ultrasonography? So as I mentioned earlier, when we're working with conventional ultrasound, we're trying to visualize fluid, right, inside the uterus, as well as the presence of an embryo. But in that context, we're limited to when there's a legit accumulation of fluid that we can go there and detect with a transrectal ultrasound, right? However, we do know that, you know, the open cows, they will start undergoing what we call luteolysis earlier than that, right? And by luteolysis, we mean the regression of the corpus luteum. And the corpus luteum is this ovarian structure here on the right. So this is the structure that produces progesterone. So this structure is required for pregnancy maintenance, right? So when a cow is open, we have the endogenous secretion of that prostaglandin that will cause luteolysis. So the prostaglandin is coming from the uterus of the cow, and it's going to break down that CL and decrease the progesterone. And what that's going to do is get that cow started to recycle again or express estrus again. I guess that's a better term. So as this concentration of progesterone will go down, the concentration of estradiol will go up. Those cows are going to show estrus and potentially be rebred in a heat detection setting or a natural service, right? But you know, the idea of utilizing the color Doppler ultrasonography comes from the fact that the blood flow to that luteal structure here, to the corpus luteum, decreases earlier than the actual structural luteolysis occur, right? So the question we're trying to ask you is, can we go in and using color Doppler ultrasound, and can we determine which cows are undergoing luteolysis by visualizing differences in blood flow to the ovary, more specifically to the corpus luteum? And this is a video with an example of a color Doppler ultrasound examination. Basically, what the color Doppler examination will do is not only visualize from a brightness mode standpoint the ovary and the structures within the ovary, but we're also going to be measuring and visualizing blood flowing through different structures. And we know that the corpus luteum here is highly vascularized. So the idea is that we can go in with that new technology and visualize those differences in blood circulation. So as I played this video here, we have two different images, one here on the right and one on the left. The one on the right is an image on a traditional ultrasound B mode, and the one on the left is an image with the color Doppler. And the color Doppler signal for vascularization is going to show up in your screen red or blue, okay? So every time you see a red or blue, that means that you have signs of blood vascularization. So initially that, you can see this is noise here because of the motion of the probe. But as we play that video here, we can see that if we look at our conventional ultrasound here, we can see that corpus luteum, kind of resembling what I showed you here. So this is an ovary with a corpus luteum here in yellow, and this is the exact same thing we're seeing in this B mode image here. So this gray structure here is the corpus luteum. And then when we look at the color Doppler here on the left side, we're trying to visualize signals of blood flow within that structure here, right? So as I play that video, you can see that this is a corpus luteum of a cow that is highly vascularized, which based on this method suggests that she will be pregnant at that stage. Even though we cannot visualize the embryo or fluid inside the uterus, by visualizing blood flowing into that specific structure, we would recognize cows that are not undergoing luteolysis, and more specifically, we'll be able to recognize cows that are in fact undergoing luteolysis and determine those cows to be open, okay? So you can see here, I have differences in CO area, but not only that, differences in blood flow measured through color Doppler ultrasonography at different stages of gestation. And you can see that there's a significant decrease in the signals for color Doppler blood flow around day 20 of gestation, right? And not only that, there's some research, this is all research performed in Brazil out of Guilherme Pugliesi's group, showing that there's actually a greater correlation between the percentage of CL that is vascularized through color Doppler ultrasonography examinations and the concentrations of progesterone than when we compare the concentrations of progesterone and the actual area of the corpus luteum, indicating that color Doppler ultrasonography might be a better estimate than conventional ultrasonography to visualize those cows that are undergoing luteolysis. There's research that has been done in South America trying to visualize and those differences in blood vascularization to the corpus luteum, but not only that, trying to determine pregnancy status, both in beef and dairy herds. And one thing that I want to draw your attention here is there are differences in accuracy when you look at the dairy data. And the reason for that is because there's a higher rate of false positive results in the dairy data sets. And the reason for that is very likely the fact that we observe a little bit more embryonic mortality on those dairy cows. So basically, when we're scanning those cows at day 20 of gestation, they could potentially be pregnant, but as we wait from day 20 to day 30 to compare the color Doppler ultrasound with the conventional B mode ultrasound, there's some pregnancy loss occurring that is decreasing the accuracy of those tests. Then we look at the beef data. The beef data is way more accurate with really relatively low false positive rate results. One of the things to keep in mind is that with the color Doppler, we observed 0. Here we had 0.5%, but here we have 0% of false negative results. So if you think about utilizing that technology in the context of resynchronization, it seems like it's a promising technology, right? But unfortunately, there's few data on our beef population, and this is something that we've been trying to tackle. We know that there's some physiological differences in our Bos taurus beef population here when we compare to the Bos cínicas population, in which a lot of these technologies were developed down in South America, or not necessarily developed, but validated in larger experiments. So, and not only that, there are differences in subspecies with regards to embryonic mortality, as well as characteristics of the CL when it comes time, when those corpus luteum are undergoing luteolysis, right? So we wanted to validate those results within our beef Bos taurus population. That's something we've been tackling here at UGA, and this is the experimental design for that study. So basically, we're exposing those cows to round of synchronization followed by fixed time AI. And then we go in at day 20 and 22 of gestation, and we perform color Doppler evaluation of the corpus luteum. We record videos of those CLs using the color Doppler. And then at day 25, we collect blood samples for PEGs. And then at day 30, we go there and compare all these three methods. So the color Doppler at day 20, the color Doppler at day 22, and PEGs at day 25 with the gold standard transrectal ultrasound, we utilizing the B mode at day 30 of gestation, right? Not only that, we're also evaluating pregnancy at day 100 to see if there are any differences in these parameters associated with late embryonic mortality. I'm not going to show a whole lot of that today, but I am going to present some of our data with regards to the color Doppler. So we have videos for more than 200 cows now, and all those videos are recorded. And with regards to our criteria to differentiate pregnant from open cows in both day 20 and 22 with the color Doppler, we're defining a cow as being non-pregnant, a cow that either doesn't have a CL, right? Or a cow that do have a CL, a corpus luteum, but that corpus luteum had 25% or less of its area being vascularized. Okay, and another layer in that criteria is the fact that if we had a corpus luteum smaller than 2 centimeters, the cow is also considered non-pregnant based on these criteria here. And again, we utilize this criteria and we compared with our pregnancy diagnosis performed at day 30 with conventional ultrasound. So this is, again, the example of our color Doppler ultrasonography examination here. So here is just noise at this point. Then when we grab the ovary, you can see the CL, and you can see quite a lot of vascularization in that CL, right? So this will be a cow that we will consider pregnant based on that criteria. And here we have a cow that we will consider open based on the previously mentioned criteria, okay? And what I want to draw your attention here that I brought the video with the cow that we consider open that had the highest vascularization. So just to serve as an example, but you can see that there's some pretty clear differences with regards to the amount of color Doppler signal within the luteal structure, right? We get a little bit of signal in the periphery of that CL, and a lot of, we got some in a specific signal here as we're moving our probe in that ovary. But within that CL, there's little to no signal. So in that context, we'll be considering that cow open, right? So when we compile the data and we evaluate, so this is what we call a receiver operating curve. So don't worry too much about that. Focus here on the right. So here we have the accuracy and the percentage of false positive and false negative results for the PEGs that we collected at day 25, at day 29 as well. But what I want to draw your attention here is to our results for the color Doppler. So similar to what is observed in the data in the Boston animals, our false negative rates using those conservative criteria are 0%, meaning we're not calling any pregnant cow open, right? Because ultimately through that color Doppler ultrasonography, we're trying to identify the open ones, right? Rather than diagnosing the pregnant ones, right? And we have a pretty decent accuracy with 86% at day 20 and 90% at day 22. And all those, that decrease in accuracy was coming from false positive results, meaning that there are few cows, in this case here, at day 20, 14% of our cows, we're calling them bred, but at day 30, when we're running them through again, they're coming out as open with the conventional ultrasound. When you look at day 22, that number decreases to 9%, okay? And one of the things to think about, is what's driving this? Why it's not 100% accurate, right? When you go in with the color Doppler ultrasonography. And there are a few things that could be happening here, right? And we don't really know the answer. But one of them is the lack of synchrony. We could have some of these cows that fail to respond to the synchronization protocol. And then whenever we're running them through at day 20, day 22, they are at random stages of their cycle, so we're not scanning them at that specific time where luteolysis is occurring. Not only that, but what I think it's more likely happen is that the vast majority of those cows, they are in fact pregnant at day 22. and at day 20 of gestation. But when we run them through at day 30, we know that there's quite a bit of loss that occurs between day 20, 22, and day 30. So that pregnancy loss is likely driving some of those false positive results. So, okay, from a producer perspective, right, from a practical standpoint, so is there a potential for the use of color Doppler ultrasonography in the near future? And so this is the compiled data for our study here, and here we have the results for our conventional B mode ultrasound. So we had a 53% pregnancy rates in the study. Okay, so we had 53% pregnant cows and 40% of them open. Out of the 40% open, when you think about the total number of animals, only 14% of the total number of animals represented cows that we misdiagnosed. That meaning that we call them bred and they were actually open on day 30. Okay, so that means that we were able at day 20 to detect 71% of the non-pregnant cows, and at day 22, 80% of the non-pregnant cows with this color Doppler technology, utilizing that specific criteria. And as we think about potentially utilizing that in the industry, right, remembering that we have 0 false negative results, here we have the heat distribution of cows after timed AI, but I want to draw your attention here to the cows that do not show heat. So basically, if we go there and go in the literature and you know, look at studies where folks were there for 10 days, going there mornings and afternoons, trying to detect those non-pregnant cows to rebreed them. They were failing to detect about 31%, about 30% of them, okay, in a heat detection setting. Meaning that, you know, we're getting some results that are pretty similar to what you would get if you go there and detect those cows for 10 days, every day, mornings and afternoons. So I consider that somewhat of an encouraging result with regards to detecting non-pregnant cows. But ultimately, what's going to drive the potential utilization of that is the actual fertility that we're going to be seeing when we start resynchronizing those cows really, really early, right? And that's something that's our next step from a research standpoint, is to characterize that. What kind of fertility can we get when we're utilizing color Doppler ultrasonography in the context of resynchronization? Okay, and not only that, how those false positive results will play a role in the overall number of AI pregnancies we can generate. We're going to be able to generate pregnancies early, but how many more pregnancies are we going to generate, and how many pregnancies are we going to not generate because of those false positive results? So these are things to keep in mind as we move forward, you know, and we keep working on this, trying to establish resynchronization programs that combine Doppler, that utilize color Doppler ultrasonography. So in summary, there are several methods available that work well, right? So we talk about rectal palpation, ultrasound, blood test. Not only that, but we know that, you know, all these tests are based on the literature that is out there, and also our experience in the field, they work really well, right? So, you know, if you go there and work with a veterinarian and figure out what works best in your operation, you can choose any of these methods and you know, you have a successful result as long as you're doing things right, right? And you can actually rely on these techniques and in these methods as well. So not only that, we can utilize pregnancy diagnosis strategically in the context of resynchronization programs. And the idea of that is to maximize the number of AI pregnancies. And not only that, as we think about the future of pregnancy diagnosis and potentially what kind of technologies are we going to be utilizing in the future, are we going to be able to potentially predict embryonic mortality? We don't know, potentially. Maybe in the future, we might develop techniques in which we can more accurately predict embryonic mortality. Not only that, is color Doppler ultrasonography a feasible alternative to resynchronize estrus and increase the proportion of pregnancies that we're generating through reproductive biotechnologies, right? So we're talking about both artificial insemination and embryo transfer. So in South America, folks are utilizing this commercially, but we need to validate those results here and see if it makes sense. There's a lot of physiological differences. There's a lot of reasons why folks that have more Brahman influenced cattle in those parts of the world are resynchronizing cows. That might not make a whole lot of sense for us here, but this is something that needs to be investigated. So we need to try to tackle this from a research setting to see if we can come up to feasible and recommendations for producers in the near future. So with that, I'd like to thank the faculty members, as well as the students and interns and forum staff that had helped us generate some of the Doppler work, as well as the folks that have been funding our program since I got here in Georgia. We really appreciate that. And with that, Vitor, I'd like to thank you as well for the invite and turn it back to you to potentially answer some questions we might have. I cannot hear you, Vitor. Not right now.
Hello?
Yes.
Can you hear me,
yes.
Pedro?
Yes, we can hear you now.
Apologize for that. Just want to remind people to please send your questions on the Q&A tab of Zoom. And Pedro, the first question that came up early on is about the cost of an ultrasound machine nowadays. If you could comment on that.
Yeah, the cost will really vary depending on the quality of the machine, as well as what kind of features you need. From a color Doppler standpoint, those machines are still very expensive because there's not a lot of people utilizing in the field yet, so they tend to be a little bit more expensive. But with regards to conventional B mode ultrasounds, you can find machines for affordable prices, depending on where those machines come from and the brand of the machine. So usually going to be around the $10,000 to $12,000 range for a good quality image that it can generate some pretty good images and reliably detect pregnancy.
In one of your slides, you mentioned that you can differentiate pregnancies between AI and natural service. Can you comment again? How is that done, please?
Yeah, so to differentiate AIs from natural service, what we recommend is that folks go there and do, let's say, one round of fixed time artificial insemination. And then before turning the bows out, they wait around 10 to 14 days. And then whenever they schedule the pregnancy diagnosis, if you're scheduling with someone that is going to come in, your veterinarian is going to bring a conventional, an ultrasound. As long as you're below 90 days of pregnancy for the first AI, differentiating the pregnancy from 90 to 75 days, it's relatively easy. So folks can do that pretty accurately. So as long as you're below that, if you can't do that and you really want to distinguish AI versus natural service pregnancy, you use the same approach. Of course, wait 14 days before you turn your cows in. But you can go in around day 30 and perform a first pregnancy diagnosis, either through ultrasound or blood test. And then at the end of the breeding season, you can go there and perform a second one. So for those of you that have a long breeding season, that's usually the way to go. So you're going to have to rely on at least 2 pregnancy diagnoses to differentiate those.
There's a couple of questions about, are you able to determine age of pregnancy on using the PAG, the blood tests, pregnancy diagnosis?
No, the commercially available blood tests, they are a yes or no type of test, at least as far as I know. The ones we work with, they're always a yes or no test. There might be ways to, there's some substantial differences in PAGs when you compare early versus late gestation. But, I'm not sure if anybody's offering that service, and I don't know if anyone has evaluated how accurate would those tests be with regards to estimating the age of the pregnancies based on solely the pregnancy-associated glycoprotein. It's more a qualitative approach, either yes or no.
There's a couple questions about costs. One specifically about the cost of the blood tests, and another one about comparing the costs of using just palpation, ultrasound, or using the blood tests. Could you comment on that again?
Yeah, the cost will vary, right? So whenever we bring someone in to perform those pregnancy diagnoses, it will depend on how far you are from them, as well as how many animals you have. So the fewer animals you have, the more expensive per animal those tests get. With regards to the technique itself, there's so much variation with regards to price within each one of these techniques that they pretty much overlap. So it's basically, to summarize, regardless of the technique you use, it ultimately, there's some other factors that are going to be driving that price rather than the technique itself. And these factors are, how far you are from who's coming to preg check, and also how many animals do you have.
There's two questions that are a little bit similar. So I'm going to try and combine them both. One is if there's more embryonic mortality before 30 days, why do you really want to go early to detect pregnancy if you're going to be, still going to have a higher or high possibility and probability of loss?
And that's a great point, right? And I think that's one of the things that we're going to try to answer with the Doppler is it really worth it to go that early, considering the amount of pregnancy loss that is occurring between day 20 and day 30? Because ultimately the rationale of going earlier is, we want to get more cows bred early to, we know that there's a lot of benefits associated with that, right? Not only from a calf performance standpoint, we know that the calves, they're born early, they're heavier at weaning, the replacement heifers breed better, they stay longer in the herd, but also that cow that calves early, they also rebreed better in the subsequent breeding season, right? So every day we gain, there's a value to that, right? So on a pound of calf basis, every day we, for every day that we gain in conception, for a given cow, you're likely going to be weaning a calf from that cow that is going to be close to 2 pounds heavier, right? Between 1.5 and potentially 2 pounds heavier. So every time we focus on getting our cows bred earlier, we're ultimately going to be improving the performance of our calf crop. So that's the rationale of trying to go as early as we can. But what we need to figure out, and that's within our beef population here in the United States, is based on our rates of embryonic mortality and the amount of cows that we can detect as open, as well as the fertility of those cows in a Doppler setting being resynchronized with a GnRH-based protocol. Is it worth it to go there a week earlier? And that's something we don't know yet, but we're gonna generate that data pretty soon.
What do you think are the chances, or what's the incidence of actually like a technician or veterinarian causing an abortion when you preg check at day 30, for example, with an ultrasound or just regular palpation? You think that's high enough that would justify waiting until 45, 50 days to do the first preg check?
I know that there's some data out there, particularly older data in the literature, where folks were doing, maybe relying on techniques such as the membrane slip and things like that. They saw some 1 to 2% increase in pregnancy loss. I have never seen such data with an ultrasound. I'm not sure if it's out there, but I wouldn't expect to see any difference, right? When we go in with the ultrasound, a lot of times we're not even grabbing the reproductive tract, and we're being really delicate there. So I don't think that will cause any type of abortion. And with regards to the rectal palpation, as long as you're bringing someone that is trained and that they know what they're doing, it's very unlikely that you're going to have high rates of pregnancy loss being caused by the technique itself, as long as the person knows what they're doing. That's again why it's important to bring in a veterinarian that really knows what they're doing.
Exactly. So there's a specific question about, so if the cow loses that pregnancy around 30 days, or right before that 30-day pregnancy diagnosis, how long does she take to get pregnant again? Are you aware of any those numbers?
Yeah, so that cow that is undergoing early embryonic mortality, a lot of times she's going to start cycling right after she experienced embryonic mortality, right? So she's going to undergo luteal regression. She's going to absorb that pregnancy that it's there. She's going to start cycling again. So from a timing of in which that cow is going to be pregnant again, that's probably going to be really soon. So we have more cows having early embryonic mortality, but they're more likely to rebreed, right? When you think about our population, our cows, they're experienced late embryonic mortality. So those cows are less likely to rebreed because they're going to start cycling again once they, they're clean the reproductive tract, they undergo complete luteolysis and they'll start cycling again. But the reality of it is a lot of times the breeding season is finished by the time that happens. So yeah, it depends on what type of pregnancy loss we're talking about. Usually early pregnancy loss is more likely that the cow is going to rebreed, whereas late pregnancy loss is likely different.
Last question that we have here on the tab is, can you comment on some strategies? It's kind of it could be a long answer, but some strategies to minimize fetal loss, and what's available out there, and what do we know about that?
Fetal loss.
And early embryonic loss, probably.
So with regards to strategies, there's not a whole lot out there with regards to early embryonic loss. With some of the early embryonic loss, right? One of the things that we know is herd health, right? So we know that there are several pathogens and infectious diseases that will cause abortion during different stages of pregnancy, ranging from early pregnancy, mid pregnancy, and late pregnancy. So having a good, herd health program that's a preventive measures for both early and late embryonic mortality. Some of the things that we know that influence embryonic mortality are, for example, we know that the rates of pregnancy loss between day 30 and day 100 of gestation is influenced by estrus expression, right? So cows that express estrus, they have fewer pregnancy loss between day 30 and day 100 of gestation, for example. We also know that there are sire-related factors, and we know that some bulls will have more pregnancy loss, particularly late embryonic loss, than others. So, as we think about, moving forward with regards to understanding bull fertility, that's something that, folks are trying to address as well. And there are some things we can do to potentially, with regards to early embryonic loss and things that we know that alter embryonic development as well as diet, right? So there's got a lot of good research out there that shows that if you have really short nutrient restriction after AI, we change that the environment of the reproductive tract, and that influences embryo development and pregnancy establishment as well. So, for example, if you think about folks that are developing heifers in a feedlot, and then when we turn those heifers out for grazing, there's a lot of good research out of South Dakota that shows that those factors, will influence the quality of the embryo and potentially change pregnancy rates. So there are a lot of things that we know that influence that, even some specific supplements. There's some research there's actually being presented here with Dr. Cook's group with supplementation of specific fatty acids that are also helping embryonic development and can potentially mitigate early pregnancy loss by enhancing the development of those concepts. So there's a lot of things that we're trying to do with regards to trying to understand those factors. And as time goes by, we're likely going to have more and more recommendations with regards to how we can optimize embryonic and fetal development to minimize those losses. Yes.
All right. I have one more question, so we'll do that one, and then we'll be done for tonight. What are the chances if you're bringing cows, within those 30 days after AI, for example, through the chute? Are you risking increasing the number of embryonic loss because of the stress of working cattle through the chute early on, right after AI?
Yeah, so it will depend a lot on the timing in which we're doing that, and not only that, but how we're doing that. Right? If we have cows that are well acclimated, well handled, we expect those the negative impact that we would have from that stress to be minimized or potentially completely eliminated, right? Whereas if you're in a situation where, you know, animal handling is not the best, that's something, you know, as a technician that is, or a veterinarian that is, you know, think about implementing some of these resynchronization strategies. These are things to keep in mind. You know, their operations, they're just not there. They're just not ready for that kind of handling yet. And we've seen that happen before.
All right, Pedro, thank you very much. Really appreciate the talk. Very important topic. Hopefully you convince some people to price check their cattle with some great options that we have available right now. So with that, thank you everyone. Again, this webinar and all of the webinars for this past spring are available on our YouTube channel, and were recorded and available on our YouTube channel. And we'll hope to see you all this fall during the Apply Reproductive Strategies, our annual meeting. And we'll see you all again next spring for the next webinar series. So Pedro, thank you again. Good night, everyone. And we'll see you soon.